Good afternoon, everyone. Welcome to Workshop 26, CFTR Modulation, Pushing New Frontiers. My name is Rachel Linneman. I'm a pediatric pulmonologist and clinical trialist who specializes in modulator trials. I'm at Emory University and Children's Healthcare of Atlanta. My co-chair is Dr. Gergely Lukács, professor of physiology and biochemistry at McGill University. We're very excited for the session we have today. I do receive grant support from the CF Foundation, NIH, and Vertex, as well as consulting support from Vertex for modulator trials. Dr. Lukács has no relationships to disclose. As we've heard in the plenary so far, over 90% of people with CF have CFTR variants that may respond to currently available, highly effective modulator therapies. Yet complete functional restoration of CFTR activity has not been reached. At the same time, some people with CF do not have a strong clinical response to current CFTR modulators, or they have intolerable side effects and need additional options. In addition, individuals with rare CFTR variants may respond to modulators, but do not have a clear pathway to gain medication access. And cost remains a significant barrier to drug access in many countries. So novel CFTR modulators, add-on therapeutics to potentiate modulator efficacy, and innovative in vitro theratyping models are needed to meet clinical needs. And today's workshop will highlight research that addresses these topics. Our format today, we have 5 speakers. Each will have 15 minutes for their presentation, and then we'll have 8 minutes for discussion and Q&A. We encourage you to submit questions through the app. I think everyone knows how to find it now. Go to our session and to questions. But you're also welcome to walk up to the front of the room, and we can turn the mic around so that you can ask your question in person. If you're sitting in the back of the room and you'd like to move forward, please feel free. And I'm going to turn it over to Dr. Lukács.
Good afternoon. So I will give you a very brief overview of the 5 speakers whom we have the fortune to have. So the first speaker will be Dr. Harblatt, and he will provide an overview of a novel triple combination corrector preclinical development that can lead to a fully folded and Y-type-like functional delta 508 CFTR by stabilizing not only the TMD domains, but also the MBD1 domain, which is pretty unique, that has not been accomplished previously. The second and the third talk by Susan Cross and John Brewington will discuss results using intestinal organoid as a model system and primary nasal epithelia to identify rare CF mutations that are susceptible to ETI and the triple combo originally developed by Proteostasis Therapeutics. Finally, the last 2 talks by Charlie Bengtson and Amanda Centore will present avenues to reverse the deleterious secondary consequences of CF mutations, namely the elevated TGF-beta level and the lipid metabolism imbalance that contribute to the CF phenotype and the reduced drug susceptibility. So this is just quickly the summary slides of each talk. This is the MBD1 stabilization, which has not been done before. This is the second talk, which will use intestinal organoid for drug selection, and then the nasal epithelial model system. And finally, the 2 talks regarding the secondary side effect, TGF-beta diminished modulator effect, reversal by pharmacological means, and the lipid metabolism reversal by fenretinide trials. So without further ado, I would like to call upon Dr. Harblatt. The stage is yours.
Thank you so much. So we had a slide catastrophe right before this session. So let's see if this actually works. You say that now. All right, are we good?
Yep, we're good.
Okay, I'd like to start by thanking the session's moderators, Rachel and Gergely, for selecting our work for presentation. This is really the first time that any of this has been, are we not able to hear? Okay, thank you, Nana. The first time that any of this has been presented publicly, and so I hope you find it as exciting as we do. So here's my disclosure slide. I'm kind of more used to non-disclosure slides, but here we go. All right, so as a reminder, and there's probably no one in this audience that needs it, so CF is a severe genetic disease. It's caused by loss-of-function mutations in CFTR, and while there are hundreds of different CFTR mutations that cause CF, the vast majority of people with CF have a specific mutation, and that is the delta F508 mutation. So this mutation causes CFTR to suffer a severe folding defect. The protein is trapped in the ER. It doesn't get to the surface of the cell where it needs to be to function as a channel. And in the last 3-4 decades, we as a field, and I speak not just for me, but for other people since I wasn't part of the field then, have learned a lot. So not only have we uncovered the cause of CF, we now know that pharmacological restoration of CFTR function can be disease modifying in many patients. While this is great, we also know that for most patients, our existing therapies do not restore CFTR function to wild type levels, and for us, this is really the goal. So to be able to provide patients with more benefit, we really need to understand the nature of the delta F508 defect. How does this mutation cause delta F508 CFTR dysfunction? So we know that delta F508 leads to the loss of phenylalanine at position 508 in the first nucleotide binding domain of NBD1. And work from Christy Brouillette and John Hunt many years ago showed that in the presence of this mutation, the NBD1 domain unfolds irreversibly at physiological temperatures. But that's not the whole story, because we also know from Gergely's work and Phil Thomas's work with insights from Isabel Calbo that the last loss of this phenylalanine leads to a weakening of the interface between the NBD1 domain of CFTR and ICL4, the fourth intercellular loop of its second transmembrane domain. And together, these two defects are really what drive dysfunction. So fortunately, we know that if we can impact these two different factors, we can robustly correct, if you will, delta F508. And what I'm showing now is work from Pat Thibodeau. And Pat Thibodeau, if you're in the audience, thank you again for letting me use this. I should have also thanked Gergely because he was in the lab at the time, but there you go, I forgot Gergely. Can I use your, yeah, okay. But anyway, here what we're looking at is the effect of second site suppressor mutations on delta F508 maturation. And I'd like to point out something kind of interesting about assays like this. The most sensitive measure of protein folding is really the ER quality control machinery in the cell. If a protein is well folded, the ER knows it. And in the case of CFTR gets out of the ER if it's properly folded. It goes to the Golgi, it's glycosylated, and you can see it on a Western band. You can see it on a Western blot, and by this higher molecular weight species. All right. So what we can see here is I. I is we fixed the NBD1 ICL4 defect, partially perhaps, with the R1070W mutation. What you can see is an increase in the level of that apical glycoform. The protein is better folded. If we use other mutations, and Pat used 3 here to stabilize NBD1 in the context of full-length CFTR with the delta F508 mutation, you can see an increase in this apical glycoform. But interestingly, if you do both at the same time, you have a robust increase in this band C. The protein is much better folded, and you can see the same effect in terms of the trafficking of the channel, the function of the channel, the half-life of the channel, et cetera. So this has really been our roadmap. And it's really supported by work by lots of different labs, Ineke Brockman's lab, Jack Reardon's lab. Lots of people have shown the same thing, that we have 2 key drivers of dysfunction, NBD1 instability and interface assembly defects. And if you can fix them both, you have a chance of pushing this channel to wild-type-like behavior. How am I doing on time? Good. Okay, good. All right. So despite the fact that it's been clear for a long time that NBD1 stabilization is sort of the holy grail in the field, it's also an area that's been fraught with challenge. And I just highlight this paper from Pfizer, the Hall paper from 2016. I mean, after spending 5 or so years and presumably spending tens of millions of dollars to try to identify NBD1 stabilizers, they came to the conclusion that NBD1 itself may lack the features that enable a high affinity interaction with a small molecule. Right? That would be disheartening to any person who read it who was interested in NBD1, but it didn't really cause us much concern because by that time we already had potent NBD1 stabilizers that worked in cells. So the work I'm describing here really leverages 12 plus years of effort. This is an effort I started at Genzyme and carried it through at Sanofi with support from the Cystic Fibrosis Foundation. And all of the CF programs from Sanofi are now contained within a company we called Sionna. So as a result of all this work, we now have a first-in-class NBD1 stabilizer that's IND-enabled and heading towards the clinic. But we also have a portfolio of additional NBD1 stabilizers that we're advancing, and I'm going to describe but one of them in this talk. But just as we know from second site suppressor mutations that you need to stabilize NBD1 and fix global assembly to really drive full restoration of function, we have programs targeting ICL4, and I'll talk more about that, and also TMD1-directed correctors that have a mechanism similar to lumacaftor or tezacaftor's mechanism. So again, our goal is to use these complementary assets in combination to reach fully restorative therapy. So I'll talk to you guys about Scion 638. So this is our first IND-enabled NBD1 stabilizer. This compound is a bona fide NBD1 ligand. And here what we're looking at are SPR data that show that the compound binds with high affinity to NBD1 and one-to-one stoichiometry. And we assessed thousands of such ligands over the course of many years. We heavily leveraged structural biology. We solved the NBD1 compound crystal co-structures for more than 150 compounds. This is a massive effort. And once these things bind to NBD1, they increase its stability. And what we're looking at here is the unfolding and aggregation of NBD1 as a function of temperature. So this is an assay that probably everyone in this room has conducted, though you may not know it. Every single time you scramble an egg, you're looking at the temperature of aggregation. So here what we're doing is we're heating up NBD1 until it scatters light as a consequence of aggregation. And so what you can see in that pink curve there, that's the unfolding and aggregation curve for a form of delta F508 NBD1 that's tractable to work with. And you can see it's less stable than the light blue line, which is wild type NBD1. But in the presence of Scion 638, the stability of this protein is dramatically increased by roughly 10 degrees. And this is an important feature. This is what we were hoping we could find. And it's also a feature that's unique to NBD1 stabilizers. If we look at TMD1-directed correctors like lumacaftor or tezacaftor or our own TMD1-directed correctors, or the data is not shown here, ABV222, there's no impact on NBD1 stability. If you look at what we call ICL4-directed correctors like alexacaftor, our own ICL4 correctors, other ICL4-directed correctors that have been in the clinic, also no impact on NBD1 stability, nor do we see an impact with CFTR potentiators like ivacaftor. So we can now step away from looking at NBD1 in a test tube and look at CFTR, full-length CFTR in a cell. And we can use, again, CFTR maturation as a way of seeing whether or not the cell thinks the protein is folded or not, because it's better at determining that than we are. So unfortunately, there's a lot of data on this Western blot, and so I tried to make it a little simpler by putting some boxes around, and I'm not sure I succeeded. But what you can see here is what we're looking at is the addition of an NBD1 stabilizer in a whole bunch of different contexts to cells, in this case CFS-ME cells expressing delta F508 CFTR. So from left to right, we're looking at the application of NBD1 on untreated cells, and you can see an increase in band C. Then we're looking at the effects of a TMD1-directed corrector at its Emax, tezacaftor at its Emax. You can see a very small amount of C-band. And again, this is a complementary mechanism to NBD1 stabilization. When you stabilize NBD1, and again, this is totally recapitulates second site suppressor mutations, you see a robust increase in the amount of the mature protein. Something similar is seen when we combine NBD1 with Tez-Iva or tezacaftor and alexacaftor at their Emax concentrations, or the combination of tezacaftor, alexacaftor, and ivacaftor. In each case, we wind up with a robust increase in C-band. So, we also see the same thing by CFTR trafficking. And if you're interested in the data, you can refer to poster 662. We have some trafficking data there that shows the same synergy. But, we're really interested, of course, in the impact this has on CFTR function. And so here what we're doing is looking by EFIS at CFTR current in CF patient airway primary cells grown at air-liquid interface. And there's an awful lot of data that has been consolidated into this one graph. So this is an experiment that involves 9 CF-HBE donors who are delta F508 homozygous and 8 wild-type HBE donors. Each donor was run in 6 to 8 replicates. And what we're looking at is what's the effect of Cy638 if we add it on top of modulators that have a complementary mechanism that impact in a positive way global assembly. And you can see there's an increase in the CFTR current now reaching wild type. But our goal is really to come up with combinations of proprietary modulators that can give people full restoration. And so what I'm showing here are a couple of examples. On the left, again, what we have is a Western blot where we're looking at CFTR maturation. And we have biological replicates in each of these 2 lanes. So the first 2 lanes are untreated. Then we're looking at the effect of lumacaftor at its Emax, and you can see a fairly subtle increase in the amount of C-band. Then we're looking at a compound Scion 109, which is in IND-enabling studies now, and it also has a relatively modest effect on C-band and maturation. The next compound is a compound we call Scion NBD1A, at least for this purpose. It has a much, much more robust effect on CFTR maturation. But again, if you combine these 2 mechanisms that are complementary to each other, ICL4-directed correction and NBD1 stabilization, you have robust synergy and a really hefty amount of C-band that in this experiment is greater than what we see in wild-type cells. If we again look at function in CF-HBE cells, here we're looking at the effects of Scion NBD1A by itself compared to the Emax effect of tezacaftor, elexacaftor, and ivacaftor, and they're roughly the same. If you then take an NBD1 corrector like Scion NBD1A and you add an ICL4-directed corrector, a TMD1-directed corrector that, again, for this talk, we're calling Scion TMD1, or both of those, the function is pushed up into the wild-type level. So again, this is exciting for us because it does seem to be a path to full restoration. So again, anchored by NBD1, our goal is really to advance these complementary modulators to really deliver therapies with potentially greater efficacy to patients with delta F508 and other responsive mutations. So we have our first compound through IND-enabling studies. We have an additional compound in IND-enabling studies and numerous compounds that we expect to advance to candidate in the near term. I looked at this acknowledgement slide prior to this talk, and I have to say it's completely insufficient. So the people that I've listed here either directly contributed to figures or provided important comments on the presentation itself, which improved it today. But there were literally 100 plus former colleagues from Sanofi and Genzyme that made key contributions to this work. And I'd like to highlight Dave Stepp in particular, since his SPR data we used here. I'd also like to shout out to Niall McCarty and Bruce Stanton, who've served as, and Gergely, who's at various points have served as consultants to us, and to Bob Bridges, who is not here today, but who provided us a lot of help on EFIS. Thank you.
Thank you. The talk is open for questions, so please. If there is no question yet, I will ask the first one. So Greg, regarding the channel open probability, do you have any data?
Speak into the mic.
So the question is whether the open probability of the rescued delta 508, the channel,
yeah.
Is it?
So we don't show any of the data here, but from other data, we have reason to suspect the open channel probability becomes wild type-like as a consequence of NBD1 stabilization in the absence of a potentiator.
So, okay, so the next question, which I have the last, and then I give it over to the audience. It's beautiful rescue efficiency, what you have. The distribution of the channel between the endosomal compartments and the plasma membrane preserved or it may be altered upon?
We believe it's preserved. We believe it's wild type-like in that when we look at the trafficking of the channel, it's normalized, its function is normalized, its maturation is normalized. Every element that we've looked at, and we looked at many, now the protein looks like wild type, fully like wild type.
Great, thank you. Please come forward and use the microphone. I think
everybody gets more steps in this way.
So, in your Western blot, you showed a quite marked detrimental effect of ivacaftor on rescue of CFTR. Do you think we will need potentiator in the presence of such effective correctors?
No, we don't.
Okay.
We don't. And again, I think it's in part because the behavior in the presence of an NBD1 stabilizer becomes normal.
So that's truly amazing persistence in the face of what I'm sure were many discouraging things in people. Anyway, so it's easy to know you have an NBD1 corrector via the thermal shift assays that you showed. How do you know it's an ICL4 corrector? And then the second thing is related to that, actually, is that knowing the structure and how Alexa works, I would have expected the lumicaftor site to have a stronger interaction with your NBD1 corrector than the Alexa site. I think the data show that it's exactly the opposite, that the Alexa site synergizes with the Luma site, with the NBD1, and the Luma does not.
No, they both synergize about equally. And in terms of your first question, which is how do we define these different categories, in part it's from work we've done with mutation profiling, where we'll look at panels of different missense mutations across the channel and look for, because this is, we ran some massive screens and we needed ways to decide what were real correctors and what weren't. And so we looked at massive panels of different mutations and said, okay, what do these things actually, we theratyped every compound that we could and said, okay, how do these things work? And there were some very clear patterns of compounds that had effects on mutations within ICL4, that these compounds then, when you would test them in combination, were non-additive. And so it allowed us to build up a data set where we could essentially bucket compounds by mechanism, even in cases when we didn't know precisely what the binding site was.
Hi, yeah, great data. So have you looked, so normally in these physiological or in these in vitro assays, we use supraphysiological forskolin concentrations. Have you looked at the EC50 for forskolin and compared to wild type also?
Yeah, we have done forskolin titration assays historically with some other compounds, and the forskolin dose response looks like wild type with these compounds. Yep.
Yeah. Okay.
Yeah, thanks.
No more questions.
I think that's all the questions.
Thanks.
Thank you.
Thanks very much. So the next talk will be given by Susan Kroos. She is a PhD student at University Medical Center Utrecht, Netherlands, in the Beekman's lab. And the title of her talk is Triple Combination Therapies for the Rescue of Rare CFTR Mutation of Intestinal Organoid. Please.
Hello, I am Susanne and I'm a PhD student from the Beekman Lab, and I am going to present to you our research project called Triple Combination Therapies for the Rescue of Rare CFTR Mutations in Intestinal Organoids. So here you see an overview of treatment eligibility for people with cystic fibrosis. And currently the VX triple therapy, VX445, VX661 and VX770 is indicated for CF patients aged 6 years and older who have at least one F508del mutation. And this results in 77% of CF patients being eligible for this treatment. Also, 13.6% of CF patients have gating mutations, and they are eligible for the VX770 treatment. And this leaves 9.4% of people with cystic fibrosis not being eligible for CFTR modulators. And this percentage varies a bit between countries and continents. And also this 9.4% of CF patients are characterized by having very rare mutations and by having a high heterogeneity of these rare mutations. And in our project, we focus on finding treatment for CF patients with rare mutations. And we do this by investigating a rare mutation response to the VX triple therapy and to another triple therapy with a different mode of action. So the VX triple therapy includes a potentiator, VX770, which increases CFTR channel open probability. It also includes a corrector called VX661, and correctors improve CFTR trafficking to the cell membrane. Then it also has VX445, which has both corrector and potentiator activity. And then there is another triple therapy available for research, which includes a potentiator called PTI-808, a corrector called PTI-801, and an amplifier called PTI-428. And as far as we know, amplifiers increase CFTR mRNA stability by binding to another protein called PCBP1, which binds to the CFTR mRNA. And because the PTI triple therapy has an amplifier, it has a different mode of action. And therefore, it would be interesting to see how differently or how comparable these two therapies function. So we look at rare CFTR mutation response to these two therapies, and we also want to compare these two therapies. And therefore, the goal of our project is identifying rare CFTR mutations that are responsive to triple CFTR modulator therapy in patient-derived organoids. And our approach is to compare the two therapies on a functional level using the forskolin-induced swelling or FIS assay, and on a protein level by Western blot. Then in the next few slides, I want to explain you the FIS assay, and I want to tell you more about the organoid lines that we chose for this project. So when you stimulate healthy wild-type intestinal organoids with the cyclic AMP agonist forskolin, the CFTR channel is activated, which leads to fluid influx into the organoid, into the lumen of the organoid, resulting in the organoid to swell up like a balloon, like this. However, when you stimulate CF patient-derived intestinal organoids with no or very little CFTR function, the organoids do not or barely swell, as you can see here. But when you stimulate these same organoids with forskolin, and the CFTR function is restored by adding CFTR modulators, you can see that the swelling is restored again. And by measuring this swelling, this tells us something about whether a patient may or may not respond to certain CFTR modulators. And we quantify this swelling by staining the organoids with calcium green, and by using a confocal microscope that takes images of the organoids every 10 minutes for 1 hour. And because we stain the organoids with calcium green, we can measure the square micrometer of green signal over time. And after that, we take the area under the curve, which gives us one value with which we can make a bar plot. Then for our project, we chose 90 CF patient-derived intestinal organoid lines, of which 45 harbor rare mutations. And at the time that we chose them, these patients with these rare mutations were not eligible for available drugs. And they were also not allowed to have 2 class 1 alleles. And then we performed CFTR functional characterization using the FIS and CFTR protein characterization by Western blot. Then we also chose 45 organoid lines that harbor at least one F508del mutation, and also either another F508del mutation, a stop mutation, or a missense mutation. And these lines we use to create reference values because we want to map the range of response of these reference values. So, for example, patients that harbor the F508del stop mutations are the lowest responders to the VX triple therapy who are eligible for this therapy. And of course, between these patients, there is a range in how well they respond to this therapy. So there will be an average response, there will be a patient who is the highest responder and a patient who is the lowest responder. And we want to map this range using the FIS and then see how comparable this is to the swelling results of our rare CF mutation lines. Here I want to show you our first FIS results that we have so far. So on the x-axis, you see 21 CF patient-derived rare mutation organoid lines. And then on the y-axis, you see the swelling as AUC, and it's normalized for residual function. And then for each organoid line, you see its swelling response to the VX triple therapy in orange and to the PTI triple therapy in green. And then the dotted line shows the average response to the VX triple therapy of 2 reference lines. And we found that 7 out of 21 lines respond well to the VX triple therapy. And we also saw that often the lines that respond well to the VX triple therapy harbor the mutations A46D or N1303K. And we also saw that 2 of the organoid lines respond well to the PTI triple therapy. Furthermore, we also saw that there were 3 lines that already showed swelling before we started measuring. And this phenomenon we call pre-swelling. And I would like to explain that further. So during the FIS assay, we incubate the organoids with correctors for 24 hours because it takes time for the correctors to do their work. And then right before we start measuring, we add the potentiators and forskolin. And we add the potentiators right before we start measuring because the organoids can start swelling immediately after we added potentiators. So here you see images of the 3 lines that I showed you in the previous slides that show pre-swelling. And you see that when they are treated with DMSO, there is no pre-swelling. But when they have been treated with VX661 and VX445 for 24 hours, right before we start measuring, you see that there's already swelling. And this is because, as you might remember, the VX445 compound has both corrector and potentiator activity. And since it has corrector activity, we have to add it 24 hours beforehand, but with the risk that the organoids already start swelling because it also has potentiator function. So for these 3 lines, we underestimate the effect of the VX triple therapy. Then I would like to show you our Western blot results. So here you see a Western blot where mature CFTR protein band, also called C-band, stained for mature CFTR protein. And it's also stained for immature CFTR protein, which is called B-band. And you see this staining for a wild-type line and for a rare mutation line after treatment with DMSO, the VX triple therapy, and the PTI triple therapy. And you see that after both VX triple therapy and PTI triple therapy, there's quite a big band of mature CFTR protein. However, when we look at the swelling results on the right of this same line, you see that after PTI triple therapy, there isn't much swelling, even though there was a big protein band there. And this we see in other Western blot data as well. So here you see a Western blot for 2 other rare mutation lines. And here you see that after PTI triple therapy, there is even a bigger mature CFTR protein band compared to after VX triple therapy. However, when we look at the swelling data of these 2 lines, you see that the swelling after PTI triple therapy is very low. And then we also correlated protein level with the swelling results after VX triple therapy and after PTI triple therapy. And we saw that after VX triple therapy, there is a correlation. But after PTI triple therapy, we see that the swelling does not correlate with the protein level. So to conclude, individuals with rare CFTR mutations could benefit from triple modulator therapy. And we saw that lines often respond better to the VX triple therapy compared to the PTI triple therapy. And the lines that responded well to the VX triple therapy harbored often the A46D or the N1303K mutation. And on protein level, we saw that after PTI triple therapy, there was, we often see high CFTR protein levels, but this does not correlate with the CFTR function. And we think that the corrector and amplifier of the PTI triple therapy does cause there to be more CFTR protein, which is really exciting. Only we think that the potentiator does not work well enough to also show this on a functional level. So in future, we want to try, we want to combine other potentiators with the PTI triple therapy to see whether we can improve CFTR function. And we also want to characterize the contribution of the amplifier on a qPCR and on Western blot level. And we want to investigate the role of the PCBP1 protein to which the amplifier binds. And we want to work on an assay to tackle the pre-swelling. So I would like to thank everyone from the Beekman team, with in particular Juliette Lefferts, Natasha Nieuwenhuizen, Paul Niemuller, and Thibaut Verburg, who are working very hard on this project.
Thank you for the talk. So the talk is open for discussion. Any questions?
I'll start us off. For the patients in your studies that did appear to respond to either modulator combination but are off-label, what is the next step for them?
So the next step is to talk about this, show our results to the clinic, and then decide whether they can have the therapy or not.
Another question from here. Would you mind speculating why the channel, which is rescued very well, not functional?
Yeah, I guess the potentiator doesn't work well enough, but I don't know why.
Yeah, that's one possibility. Maybe. Yeah. So. Any other question? Please come forward.
Thank you. So obvious question. When you were seeing protein expression and mature C-band, you were seeing in the overall crude cell lysate, right? Do you plan to look at the cell surface protein, like i.e. whether the channels that are getting matured and glycosylated are actually going to the cell surface, and therefore the disconnect that you see in your result between the functional and the protein expression, whether that can be explained by the cell surface protein expression?
Yeah, we have not looked at that yet, but that's a very good idea. Yeah.
Any other? Please.
Did you happen to look at the expression of other channels? For example, ENAC?
No.
That may be affected by... If she looked at the expression of other ion channels, given that PTI 428 may not be specific for CFTR.
Yeah, no, I have not looked at that.
Okay.
That's also a very good idea. Yeah.
Thank you again. If there is no more question, we will move on.
Thank you.
So our next speaker is Dr. John Brewington. The title of his talk is Characterization of CFTR Modulator Response in Nasal Cell Models Derived from Patients with Rare CFTR Variant. He's an assistant professor at Cincinnati Children's Hospital. pediatrics pulmonology. Please.
Hey guys. I do want to say a big thank you to the organizing committee for making this workshop happen. Thank you guys for having us along to share some of our work with nasal cells. I'm John. I'm a pediatric pulmonologist, and I am in Cincinnati. I'm very excited to share this work. This has been work that's been going on for a number of years, and a large number of people have been a part of it. These are my disclosures. Now, the great thing about going in the middle or second half of a session like this is that all of my background slides have already been discussed, so this makes this part very easy. You guys have already heard the stage be set really beautifully. We have a couple of problems with the current environment of CFTR modulator therapy. One is an access gap, people who might have benefit from existing drugs but can't get them. And the other is a precision gap, where more drugs are coming down the line, like the one we heard of in our first talk here. And at some point, we'll have multiple drugs for a single individual and very limited data to understand who gets what or who would benefit the most from which drug. And one way to close those gaps is with theratyping, which again, we've talked about already. Now, I'll point out, when we use the word theratyping, you can apply that to lots of different approaches. A really powerful approach that's been used in the field to develop these drugs and for label expansion is heterologous overexpression models like FRT libraries. And that's had a great record. Today, though, I'm going to be talking about patient-specific theratyping, which we just heard another very elegant presentation about the same approach, where we take a patient-derived sample, grow a model of it, that perhaps is not as applicable to a large number of individuals, but is highly precise to the individual from whence it came. Now, I'll be talking about nose cells because I'm a nose cell guy, but as you guys just saw, it's not the only way we can get good patient-derived models. Again, the intestinal organoid model is a wonderful one. We're seeing emergence of a number of models from iPSCs that have incredible promise and power. So by talking about nasal cells, I'm not in any way suggesting those other models aren't as good. It's just what I do. Most of the models that are out there, though, capitalize either on measures of ion or fluid transport. We just heard about a model looking at fluid transport, either through electrophysiology or through swelling assays like we just saw. And we've done the same thing with nasal cells. Like others in the field, we've generated two different models based on these sort of prototypical approaches. One is air-liquid interface cultures, basing off of the bronchial cell models that I'm sure most of you are very familiar with, and another spheroid cultures based off organoids like from the intestine as well. These models have been described in the past, so I'm not going to spend much time on them today, but if you want to learn more about them, please grab me afterwards. Always happy to chat. Now, we've taken these models and we've sought out subjects who have rare CFTR variants and do not have access to modulators. Now, this obviously has changed over the years as we've done this with the emergence of new modulators into the market and into the clinic. But we've asked, can we use these as a preclinical assessment tool, similar to what we just saw, provide that data back to the patient, and potentially improve the access for patients for whom we believe there's going to be benefit from these drugs. And again, the goal is really to define the predictive capacity for these models. I will mention, whenever we get a nasal cell sample from anybody, we grow out both of these models. We use them for cross-validation, so when we see results from one model system, we can confirm we see the same thing from the other, and helps us be confident with our data. Now, over the last few years, we've expanded our reach to be able to get samples from really anywhere in the United States. The table here is not intended to be legible in any way whatsoever. It's just highlighting the centers that we've gotten samples from over just the last 2 years. It's a large number. Alphabetically, we cover almost the entire United States. If we could get Alabama, we would have fully A to Z. But we do go coast to coast here, and we have gotten a large number of cells. We've got nasal cells from over 60 different CF centers with over 500 samples for a lot of different reasons. Today I'm talking about theratyping, so I'll just focus on that. And we've had 185 different subjects that we've tested through when I did this data poll with the help of my incredible team at the end of the summer. And so this is the cohort I'll talk about. And this will be a little bit backwards. I just told you about how important it is to think about an individual, but I want to talk a little bit about that whole cohort and about what we've learned about them along the way. These 185 subjects all have a suspected diagnosis of CF or confirmed. I'll talk more about that suspicion diagnosis later. They all have at least one rare, believed to be missense mutation. Many of these are very rare and undescribed, so some of it is a bit of a shot in the dark as missense or nonsense. But importantly, all 185 of these samples and the subjects we'll be talking about were just obtained for theratyping, not for other purposes. Now, as I go through, I'll talk about people with response or no response. Just briefly, we're calling that response an increase by about 5% of wild-type function that clears the line of 10% of wild-type function. There's precedent for this in some of the FRT models. There's also good reason for this based on our experience in F508del homozygous cells treated with lumacaftor as sort of a minimal amount of clinical benefit that we were looking for. This is a number that we're consistently and constantly looking at and reassessing. And again, love to chat with anybody afterwards on thoughts on that. But that's the line that we'll use for this talk. So of those 185 different individuals, what we've seen is a little better than 1/3 fall under that responder characterization. So they have a response, they clear that 10% line. And that's to at least one drug. Obviously, some individuals have responded to multiple. When we've shared those data back, obviously with the patient's consent, with them, with their physician, and ultimately with their third-party payer, which in the United States is almost always a third-party insurer, about 2/3 of those individuals have been able to get onto off-label therapy. Now, as drugs have come out, some of those individuals have moved on to on-label therapy subsequently. But this is something that insurance companies have been surprisingly receptive to. We've also had a couple of siblings and an unrelated individual with the same CFTR variants be able to use someone else's data with their permission, obviously, to get on drug. The breakdown of responders and non-responders is shown in the pie graph, and the drug of maximal response is in the stacked bar to the far right. Just as a reminder, this is something we've been doing for a number of years. We did this before ETI was available. Since ETI has become available, that has by and far been the most common drug of maximal response, admixed with the number of folks responding to ivacaftor. When we look at the CFTR function, and we just plotted out with baseline function expressed as a % of wild type on the X here, and maximal modulated function on the Y. And this is all 185 individuals in this cohort. We actually see that the response is fairly significant. If you look at the slope of that regression line on the left, it's about a 50% relative increase to what they came with, as well as an absolute sort of percentage increase, which is expected based on how we described the group. I've also broken it out by different drugs, Lumiva, ivacaftor, Teziva, and ETI there. And if you look, there are some individuals, they're on the high left side of that graph, that really have gone from almost no function to almost wild type function. So just like we've seen in clinical trials, we have folks with high variety in their response to these different drugs. This is also, by definition, an extremely heterogeneous population because everyone involved has a number of rare variants. Now we're interested, obviously, not just in those who respond, but also in those who do not respond. And so that's 117 individuals in this cohort. As you would expect, our regression line is very tight because we defined them as not having a response. So comparing baseline to response therapy is a simple thing to do. Now, of those 117, there's 18 who have subsequently gone on label for a drug that we didn't test at the time. This is almost exclusively F508del heterozygotes who are now on label for ETI before we had it in the lab. But we do have 6 individuals who we tested for a drug, did not see a response, and are subsequently on label for that same drug. 3 of those 6 are on drug clinically. The other 3 are not for various reasons. One of them was at Cincinnati, and we did extensive clinical phenotyping. I'd love to share the data with anyone who's interested afterwards, looking at a large number of clinical outcome measures, and we actually did not see clinical benefit. It's an individual who was homozygous for a non-canonical splice variant and had very mild disease to start with. The other one, we have a little bit of clinical data, but we don't see a clear response there. But as the other individuals are able to go on drug, we'll be gathering those data and asking, is this something where we had a miss? Do we need to adjust our threshold, change the way we do things, and make sure we don't miss anyone? I will also point out, as I said in the beginning, we're enrolling folks who have a suspected diagnosis of CF. Most come with a confirmed diagnosis, but in some cases it's suspected. And if you look here to the left side of this graph, there's an awful lot of folks who had normal CFTR function at baseline. Now, my adult colleagues are going to be very comfortable and familiar with the story, which is something along the lines of, I have this 50-year-old guy who has severe bronchiectasis, one identified CFTR mutation, and an indeterminate sweat chloride, and I don't know what to do with him. I don't know if he has CF, and if a modulator worked for him, I sure would love to prescribe it to him. And it's a story we hear very frequently, and so this cohort does get skewed that way by individuals who most likely simply do not have CF. When we look at the cohort characteristics, asking are there things that we can predict response from this group, unfortunately the groups are very similar at baseline. We see the same age, same sex distribution between the groups. There is a slightly higher baseline function in the non-responders. Again, I think this is skewed by individuals who most likely don't have cystic fibrosis, but rather other diseases. And amongst responders, we do see a trend towards a higher baseline in those who respond to ivacaftor. These are usually folks who have residual function variants, and so I think this makes good sense. From a genotypic standpoint, we pulled out a number of different specific alleles or groups of alleles, asking are there differences between the two groups. And the short answer is there aren't ones that I think are actionable. We do see a larger number of F508del heterozygotes in the responder group, but this is probably a moot conversation today. These are all individuals who were tested before ETI. We're no longer enrolling people who have a single copy of F508del, so it doesn't impact our approach at the moment. We also see a larger number of individuals with a single nonsense variant. We're not enrolling folks with two nonsense variants, but with a single nonsense variant in the non-responder group. And again, I think that is a logical thing and perhaps not a surprise. And we don't anticipate not enrolling those individuals because obviously if they have a missense, we want to understand if it might have benefit. But across the other groups that we've looked, we did not see any differences. Now, the next slide I'm going to encourage you to take with a grain of salt. And I would love to talk with anyone afterwards who has thoughts on how to organize these data. But everyone always says, what are the variants that respond? And the answer I always give them is, well, it's complicated. Because unlike in an FRT model, I'm not testing a single variant anytime we run one of these experiments. We're testing two variants. Sometimes we get lucky and we have individuals who are homozygous for something, or we have someone who has a mutation in trans with a clear, unquestioned nonsense mutation. And so we can say, yeah, that variant was responsive to that drug. I've listed those variants in the top half of this table and they're confirmed, where we can feel pretty confident that there's response there. The ones with the star next to them are ones where they have subsequently had label expansion and become on label for a modulator. And there's an awful lot of those up in that confirmed category. The possible category gets a little bit more in flux. These are from individuals where we've got two missense variants, or a couple individuals on here who we tested very early on who have a couple of intronic frame shifts, which we would typically think of as non-shift, but they still had response to drug, which leaves us in a position of saying, I'm not sure which of those two variants had the response. I'll also point out we have a large number of folks in this cohort who only have a single identified variant, many of whom do clearly have cystic fibrosis. And if there's an unidentified variant in play, I didn't include them in this table at all because obviously we have no idea what's happening with that unidentified variant. Now, these are our data so far. We have a number of things that are ongoing areas of work. I just want to show you, just to stimulate discussion. Again, we'd love to chat with folks either during questions or afterwards, and also to think about other ways that we can apply these same models moving forward. One is the question that I just got to about the genotypes. How do we share these data? Currently, we share this with the subject themselves and with their physician, obviously with consent, and on to insurance company if that's the right way to go. We have shared some cohort data. The N1303K mutation that was just described, we'd previously shared some of those data in an abstract last year. These are some de-identified data for that particular variant shown here. But what we would love to do is actually collate some of these data that are being generated by our lab and by others and have a place where, as a clinician, I could go and say, is this something that I think is a responsive variant? Should I refer my patient for further testing? Is this a place to go? And so we need to think about how do we handle the variance between labs or even within labs? How do we respect the confidentiality of people who have ultra rare or private variants, wherein this could be arguably PHI? And then there's a couple of variants with inconsistent response, and in our hands, W1282X is the most common there, where some individuals have great response to drug and others do not. And so how do we handle that, other than to say, well, it might be responsive? Another thing that's frequently asked of us comes back to that adult with bronchiectasis, is can we use this as a diagnostic study? In the diagnostic algorithm, if your sweat chloride is insufficient to get you, along with your genetics, to a diagnosis, there is a recommendation for physiologic testing, NPD or ICM. But the number of centers geographically that have access to NPD is only getting smaller, I feel. Our number of referrals for NPD testing has gone up dramatically lately, and I really appreciate my lab for doing all of those NPDs. But it would be great if we had a portable assay that we could use for diagnosis. We do know that there is an association between the data we generate from these models with sweat chloride, but obviously there's lab variants, we don't know the cutoffs. Here I'm just showing a little bit of cohort data from our normative data from wild-type subjects, early data in F508del homozygotes, and a cohort that we collected along with Daniele Salinas and folks at CHLA with CFSPID, and they do move into clear groups. We would just have to determine what the cutoffs would be and how to apply that. Finally, as has been discussed already in this meeting, these patient-derived models, while low throughput, can be very useful when we think about new therapies, whether that's development or validation. And an area that I think is particularly useful in these particular models is for patients who have intronic variants or splicing efficiency variants, which are very difficult to make in a heterologous model since those are usually done with a cDNA construct. And so this is an option for that. We've also done work with a number of collaborators on editing, as have many others, and so I think this is a good platform. The one other area that's not on the slide, but that we've been working on a lot lately, and I've chatted with a few folks that I actually see in the room about this already during the meeting, is trying to get down the numbers of contaminated samples, which has been a problem for us, and is something that's very frustrating for the clinicians and for the patients. And so we've got a number of things going where we're working to try and bring that number down. So with that, I'll conclude. Hopefully I've shared with you some about the cohort level data from our nasal cell models. It's shown that they can expand patient access and that third-party payers can be receptive to that. And we can use this to help improve access, at least here in the States. There's a lot of future potential use that I think really is untapped at the moment, whether that's for therapeutic development and also for potentially diagnostic utility. Big thanks to the folks who've been a part of this, both in Cincinnati. My lab team's sitting over here. They're incredible, and they do all the actual work and keep the train running. We also, a huge thanks to anyone who's ever sent us cells. We really appreciate that. We couldn't do it without the collaborations, and obviously to our mentors and to the folks who've paid for it. And I'll stop there and happy to take questions.
Sure. I'll start off with a question. So for those patients who have the variants that you had on your confirmed list, what would you recommend to their clinicians if they're listening online or sitting here? Should they send nasal cells to you, or should they, try and use some of your data to go ahead and get approval?
Yeah, what I tell folks is if you have someone who has a variant either that we've seen response in, or you're not sure if we've seen response in, please just email me and we'll chat. I have, in a number of cases, shared de-identified data with other folks. I will tell you that insurers have not been nearly as responsive to that approach as to a letter that says cells from this patient showed this response. We've been lucky in a couple of cases that it's not been as successful, but we can at least start with that, and then if it seems like it's right for the patient and for the physician to move forward with that theratyping, then we go forward with that along the way.
Any questions? Comments?
I'll ask another question.
Nobody wants to come to the front of the room.
I know. Oh, here we go.
In the interest of full disclosure, I'm a clinician, I'm not a CFTR scientist, but I make a request, and that is something you alluded to, which is that all of these therapies are very expensive. And while this might not apply in Europe, in the United States, the insurers often are looking for any way to not pay for somebody's care. And so we have to be careful when articles are written about cells responding or not responding in some way, to leave plenty of room to say this was in this particular assay. It might not be applicable to other parts of the body. We have to be careful not to give them, to weaponize the work that will prevent us from being able to use it. I'm not trying to say we want to use this in people where it's not appropriate. I have a better sense of finances than that. But I want to have the leeway to make the decision and not to have it made for me by the insurance company.
Yeah. Yeah, and Hank, I'm a clinician too, so I completely agree with you. And you'll notice I didn't put up any genotypes that didn't respond on the slides, and that's intentional. And as we develop these models, in no way have we told ourselves or joked to ourselves that this is perfect. And if it's negative in this model, there's no way it'll ever respond, because that's just a falsehood, right? And so our goal is that if we find data that's compelling, that we think there's benefit, we want to share that and let there be benefit from it along the way. If we find data that does not suggest there's benefit, then we move on. But I completely agree with you.
And have there been any patients that participated in your study who also participated in a study of one of the other models, like the organoids or iPS cells, to know if there's a correlation?
That is a great question. So we've had a number of patients who have had gastrointestinal organoid models made in Cincinnati, as well as some nasal cells, and we've been in a long process of collating that data to look at it. We have not yet. I think, clinically, so many of our patients have organ-specific disease. It certainly raises a question of would there be some organ specificity to the models. I think the clinician in me says probably, and the scientist in me says probably not. I don't know. But it would be an interesting question to answer. Brave soul coming to the front.
One thing that comes to my mind is that being a research lab, how rigorous we are in calling what is % wild type. Giving you an example, and for better understanding for the audience here, is that If I give you a value of 10 microamp per centimeter square for a wild-type individual, and W1282X is 0.1, and if by recovery you get 1, you will say it is a 10% recovery. But then you have another wild-type individual from another lab, and he says, my wild-type is 20, so in that case, you are coming to 5% level. How rigorous we are as a research community, first to say that these are the mean plus minus SC for wild-type individuals.
Yeah, and I completely agree. This is one of the reasons why our wild-type sort of normative data we generated with our own hands using the same methodology. If you look at, there's a number of labs that are doing similar work at a really high level, and if you look across many of our labs, you'll find that there's high variance in what we define as normal, sometimes as much as 5-fold variance in what we define as normal. This is one of the things that makes it hard to collate data from different labs. Again, for us, we thought it was important that our normative data needed to be in our hands with our own models and with a reasonable number to get there. But I think that, you know, if you're going to refer to things as a percent wild type, we need to know what the denominator is. So. Cool.
Thank you again for the very nice talk, and I think we have to move on.
So our next speaker is Dr. Charlie Bengtson. He's an assistant professor and adult pulmonologist at the University of Kansas Medical Center. Didn't work. There we go.
Perfect, thank you. Good afternoon. I'd like to thank the organizers for asking me to talk today and speak about our group's work. I have no disclosures related to today's talk. And some of the work I'm presenting today has been previously published here, and you can stop by and see us at poster 672. And so there are currently two highly effective CFTR modulators that are approved, ivacaftor and then the combination of elexacaftor, tezacaftor, and ivacaftor, also known as ETI. And they produce marked improvements in meaningful endpoints in both clinical trials and real-world studies. And the most striking improvements being seen in improvements in lung function as measured by percent predicted FEV1 and a reduction in the number of pulmonary exacerbations. In pre-modulator times, airway hyperinflammation is recognized as a key component of the pathogenesis of CF pulmonary disease, and has been a therapeutic target with somewhat mixed results. And given that not every patient responds the same way to highly effective CFTR modulators, there's a lot of interest in understanding how these modulators may impact airway inflammation, and also how the airway inflammatory milieu or specific inflammatory mediators may impact the efficacy of CFTR rescue at these modulators. And one of them that has been implicated as important in CF lung disease time and time again has been transforming growth factor beta-1. It's a secreted cytokine that activates a variety of pro-inflammatory signaling pathways. Polymorphisms in TGF-beta-1 have been implicated as a genetic modifier in some genome-wide association studies, and high levels of TGF-beta-1 are known to be associated with worse pulmonary disease in persons with CF. In one such study, Tom Harris and colleagues looked at levels of TGF-beta-1 in bronchial alveolar lavage fluid in children with CF, and they found that as compared to healthy controls, children with CF had higher levels of TGF-beta-1 in their lower airway, that the levels were not affected by the presence of Pseudomonas, and that higher levels of TGF-beta-1 were associated with a worse recovery in lung function following exacerbation. And there's also evidence that prior to the introduction of ETI that TGF-beta-1 may impair wild-type CFTR and F508del corrected CFTR function. An earlier study from a group at University of Pittsburgh found that in normal human bronchial epithelial cells that the presence of TGF-beta1 consistently reduced CFTR current, and that over time, there was also reduction in CFTR mRNA levels. In the panel on the right, you'll see work from a group at UAB. They looked at CF, bronchial epithelial cells, and response to one of the earlier generation modulators, lumacaftor. We can see that the addition of lumacaftor significantly increases CFTR current, and that the addition of TGF-beta after that reduces current to near baseline levels. And, we don't have great data yet with ETI on how airway inflammation has changed. We can look back and get a little bit of an idea from those on ivacaftor. And so the part of a group in the GOAL study had sputum inflammatory markers measured. And although individual responses varied, we can see from these pre-post graphs that there really is no significant reduction in airway inflammation after starting ivacaftor. And this suggests that at least for some individuals, restoring CFTR does not fully resolve lower airway inflammation. And previous research by the Solitaire group has identified losartan, which is an angiotensin receptor blocker that has anti-inflammatory properties, as potentially beneficial in CF. We've mostly looked at its effects on the BK channel, which is an apical voltage-gated potassium channel that, along with other, CFTR and other ion channels, impacts airway surface liquid hydration. The expression of one of the subunits of the BK channel, the gamma subunit specifically, is known to be impacted by inflammation. And in CF bronchoepithelial cells in vitro, we have found that losartan can reverse the detrimental effects of TGF-beta1 on BK channel conductance that also then results in improvement in airway surface liquid volume and improved ciliary beating. And moving on to a somewhat less commonly used CF animal model, we looked at the effects of losartan in sheep. And so in adult ewes that were anesthetized, fluoroscopic images of the trachea were obtained to measure the speed of mucus moving through the trachea as a marker of mucociliary clearance. And these were first treated with CFTR inhibitor followed by TGF-beta1, and we can see that there was a sharp reduction in tracheal mucus velocity, and that after the addition of losartan, it improved to near baseline levels. And so this suggests that in the absence of functional CFTR, losartan can improve mucociliary clearance in relevant CF models. And so then came the approval of ETI. And so given the prior data on the effects of TGF-beta1 on CFTR rescue and our work with losartan and other ion channels, we hypothesized that TGF-beta1 would negate, at least in part, the effects of ETI both in vitro and in vivo, and that this could potentially be reversed by losartan. And so starting with in vitro studies, we looked at the effects of TGF-beta1 on functional rescue of CFTR in primary CF bronchoepithelial cells that were homozygous for F508del. As can be seen from the Using chamber tracing on the left and then the quantitative assessment in the middle graph, there was a significant improvement in CFTR current with the addition of ETI. And this was reduced in part by the exposure to TGF-beta1. And then also consistent with prior literature, we found that there was no change in CFTR mRNA levels with ETI, but it was significantly reduced after exposure to TGF-beta1. So then we added losartan to this treatment paradigm, and there was a significant improvement in CFTR current with the addition of losartan in the presence of TGF-beta1. And although it was not back to ETI alone levels, we then looked at other important physiologic assays. And so we looked at changes in airway surface liquid, and we found that losartan restored the benefits of ETI in the presence of TGF-beta1. And we also looked at percent mucus solids, a higher percentage indicating more hyperdense mucus, and we found again that losartan improved the detrimental effects of TGF-beta1 in ETI-corrected cells. And so to better understand the mechanism by which losartan could potentially be beneficial, we decided to look at the effect on CFTR mRNA, and then microRNA 145. So microRNA 145 has previously been shown to be antagonistic to CFTR mRNA, and it's also known to be increased by the presence of TGF-beta 1. And so we looked at levels of microRNA 145 and found that they were not significantly reduced by exposure to losartan, but were significantly increased by TGF-beta 1. And then looking again at CFTR mRNA, we found losartan did not significantly change CFTR mRNA levels. And so given this, we decided to look at other potential pathways that could be causing this beneficial effect of losartan. And so we looked at expression of other pro-inflammatory mediators which are known to be affected by TGF-beta 1, namely TNF-alpha and COX-2. Both mRNA expression and levels of secreted TNF-alpha were increased in response to TGF-beta 1 as expected, and again lowered significantly by the presence of losartan. And then COX-2, which is known to play a role in inflammation in the CF airway, and at least in children, inhibition of is therapeutically beneficial. We found that there was significant increase in COX-2 expression after exposure to TGF-beta 1, and this was reduced by the presence of losartan. And then we wanted to look to see if COX-2 inhibition would kind of recapitulate the effects that we see with losartan. And then, so using a commercially available COX-2 inhibitor, NS398, we found that it also rescued the detrimental effects of TGF-beta 1 on CFTR currents. And so in sum, this data suggests that aberrant COX-2 expression induced by TGF-beta 1 may lead to CFTR rescue by ETI, an effect which is reversed in part by losartan. So then moving into a cohort of participants or people with CF on ETI, we developed a cross-sectional observational study, and we measured change in lung function after starting ETI. We measured levels of nasal epithelial lining fluid as a surrogate for the lower airway, brushed nasal cells, and sweat chloride in these participants. As can be seen from the demographics table here, they all had expected improvements in lung function and BMI after starting ETI, and a majority of them were on modulators prior to starting ETI. And so we found that higher levels of TGF-beta 1 were correlated with less improvement in lung function after starting ETI, and higher levels of sweat chloride after starting ETI as well, as a surrogate marker for CFTR function. We also found that expression of TGF-beta 1 was correlated with expression of COX-2, similar to our in vitro data. And in sum, this suggests that while the strength of the correlation is moderate, that there is a connection with the in vitro data, and that there are detrimental effects of TGF-beta 1 even in those on ETI. And so in summary, we found TGF-beta 1 impairs response to ETI in vitro and in vivo, that treatment with losartan in vitro significantly improves the detrimental effects of TGF-beta 1, and potential mechanisms are through reduction in inflammation, specifically COX-2 expression. And certainly future research is needed to understand the interplay between inflammation and CFTR correction. Certainly there's a lot of people that helped with this, and members of the Solitaire lab specifically were instrumental in most of these experiments, and our CF research team at KU as well. And I'd like to thank our funding sources, and happy to open it up to questions.
Any questions, comments?
I can ask one. This is very intriguing data in a small cohort of patients. Losartan's already FDA approved, so it's faster to move towards clinical trials. What do you think are the next steps to work towards a clinical trial?
Yes, so that's a great question. Data not shown here, but we have an ongoing clinical trial in non-modulator eligible population, given the data about improved mucociliary clearance with the BK channel. And then also we are planning and in the process of, kind of starting the first steps in starting a trial in those on ETI.
Thank you. Yeah, I'm Jie Chen from UAB. Because we know the losartan is the anti-high pressure the medicine. So have you compared to the high blood pressure hypertension patients versus non-hypertension patients in your group? What's the beneficial? Which one is better?
Yeah, this cohort, I don't have data on how many were hypertensive. I think in general, we don't have a lot of CF patients now, that are hypertensive, but I think in the future that will be a problem. And so we haven't exactly looked at that variable, but I think it could be something interesting to look at in the future.
Okay, thanks.
That's what I was just going to add, is that we are seeing increased hypertension with ETI, so it sort of raises an interesting clinical question. How easy is it to get measurements of the TGF-beta clinically, like for, if we have patients at other sites who are not good responders to modulators?
Yeah, it's fairly simple. We use a method that's published by a group at UNC, and so basically a filter paper in the nose after a spritz of PBS, then that's spun down and run with an automated ELISA, so it's pretty straightforward.
So we do have one question through the iPad. Is losartan currently used in the clinic? And if so, what are some potential negative side effects?
Yeah, that's a good question. Certainly it is for those with hypertension. It's one of the most commonly used antihypertensive medicines. Side effects, it can cause a kind of a transient increase in creatinine, is kind of the main thing that we watch for, and changes in potassium because of that. In healthy adults, it really doesn't cause low blood pressure, even if they're not taking it for low blood pressure. And so those are kind of the main side effects we would watch out for.
Great, thank you so much. Okay, our last speaker today, I'd like to invite Amanda Chentarame up. She is a PhD student at McGill University.
Thank you. Okay, good afternoon, everyone. As Dr. Lindman said, my name is Amanda Chentarame, and I'm a first-year PhD student at McGill University in Montreal. Today, I will be presenting to you our work titled Treatment with Law 7B Complements CFTR Modulator Therapy by Improving Lung Physiology and Normalizing Lipid Imbalance Associated with CF Lung Disease. Okay, got it. Okay. So cystic fibrosis is an autosomal recessive disorder affecting more than 100,000 Caucasians worldwide. The impaired inflammatory response in CF reflects an imbalance between the initiation and the resolution phase of the inflammatory response. This inflammation creates elevated activity levels of phospholipase A2. Now, the overexpression of pro-inflammatory cytokines and mediators is due to the metabolization of omega-6 unsaturated fatty acid, arachidonic acid, or AA. While anti-inflammatory and pro-resolving mediators are suppressed due to a decrease in EPA and DHA. Calprotectin and C-reactive protein levels are also elevated, and the lipid and fatty acid imbalances are tightly linked with the functional expression of CFTR protein. We also see that the sphingolipid metabolism is also dysregulated in CF, and in the lungs of healthy CF subjects, or of of healthy subjects, very long-chain ceramides, or VLCCs, represent approximately 70% of the total ceramide pool, while long-chain ceramides, or LCCs, represent approximately 20% of the total ceramide pool. And the relative ratios between VLCCs and LCCs is dramatically affected in CF and actually worsens with age. Now, how can we target these characteristic defects that are seen in CF? Well, one way would be with LAU7B. LAU7B triggers the resolution phase of inflammation and the induction of a specific resolvin pathway. It corrects the omega-3 and omega-6 fatty acid imbalances in cells in mice and was also confirmed by a phase 1 clinical trial. The phase 1 clinical trial was double-blind, randomized, and showed that LAU7B was safe, well-tolerated, normalized lipid imbalance, and reduced oxidative stress. This was assessed over 3 cycles of 21-day treatment with escalating doses of 100, 200, and 300 milligrams per day per patient. The phase 2 clinical trial was a double-blind randomized trial and was actually recently completed. Results are currently presented or are being presented in poster number 168, if you're ever interested in looking into that more. Now, another treatment that we can look, or that we have, is triple therapy, which is composed of 2 CFTR correctors, elexacaftor, which is VX445, and tezacaftor, which is VX661, and 1 potentiator, ivacaftor, VX770, which when used in vitro are often referred to as triple therapy. Now, this treatment targets the class 2 genetic defect, delta F508, which is the deletion of phenylalanine at position number 508. So our aims. Our in vivo aim was to assess the efficacy of triple therapy, LAU7B, and its combination in our F508del, or DD, mouse model of lung disease. In vitro, we wanted to assess 3 things primarily. Our first thing being the glycosylation status of the CFTR protein in CFB41O negative overexpressing F508del cell line. Second, to look at total and membrane CFTR levels in the presence of PA14 filtrate. And third, CFTR protein function using electrophysiology of patient-derived primary bronchial epithelial cells. Okay, so we first looked at how the combination of triple therapy and LAU7B affected lung physiology in our DD mice. Now, DD mice who have not been treated or are vehicle treated have higher resistance in response to methacholine, whereas mice with treatment have lower resistance. What we see is that although triple therapy does slightly improve lung physiology compared to non-treated and vehicle-treated DD mice, upon addition of LAU7B, the improvement in lung physiology is significantly better. And from this, we can conclude that the combination therapy of triple and LAU7B improves lung physiology in our DD mice. So given the significant improvement seen in lung physiology upon combination of triple and LAU7B, we then looked into the lung histopathology to assess lung structure. Mice used in this experiment were between the ages of 14 to 20 months old, which actually represent an advanced CF phenotype. Now, what we see is higher airway hyperplasia in our non-treated and vehicle-treated mice compared to our wild-type controls. LAU7B treatment alone significantly improves airway hyperplasia in our DD mice. However, while triple therapy on its own does reduce hyperplasia, improvement is not as striking as in mice treated with LAU7B. Combination of the 2 treatments showed further improvement in airway hyperplasia and homogeneous looking lungs. In our figure G on the right, airway thickening caused by cell infiltration showed greatest improvements in the combinatory treatment group. So the conclusion is that airway hyperplasia and cell infiltration has greatest improvement in the combination between triple therapy and LAU7B. Moving forward, we then assessed metabolic markers of oxidative stress and lipids. So the 2 stress markers we assessed were malondialdehyde, or MD, which is a marker of lipid oxidation, and 3-nitrotyrosine, which is a marker of protein oxidation. For both markers, non-treated and vehicle-treated DD mice were above baseline levels, which is indicated by the dotted green line in the figures. For both markers, triple therapy alone does not normalize lipid or protein oxidation, whereas LAU7B on its own does. Now, the combination of LAU7B with triple therapy restores the imbalances of oxidative stress markers in the lungs of the DD mice. With improvements in oxidative stress levels for the combination treatment, given the known importance fatty acids play in CF, we assessed omega-3 and omega-6 levels in the lungs of our mice. The results are quite striking, given triple therapy alone, seen in purple, does not correct omega-3 and omega-6 fatty acids, meaning they are actually still deficient. On the other hand, LAU7B treatment alone does normalize both omega-3 and omega-6 fatty acids. Upon addition of LAU7B to triple therapy, enhanced correction is observed and restored to wild-type levels indicated by the dotted green line. So with the correction seen in fatty acid levels, the effect on phospholipids is also an important parameter to assess. The reason being that the composition of membrane lipids and rafts has significant impact on CFTR protein function. In non-treated and vehicle-treated mice, levels of LCCs and VLCCs are abnormal. Triple therapy alone and LAU7B alone can normalize the levels of VLCCs and LCCs, and the combination of triple and LAU7B demonstrates better normalization of VLCCs and LCCs. So lastly, we decided to assess protein expression levels under various treatment conditions. So as previously mentioned in other presentations, CFTR protein has two glycosylated forms, the B band, which is approximately 131 kilodaltons, and the C band, approximately 160 kilodaltons, which is the mature functional CFTR that has passed through the Golgi apparatus and has translocated to the cell membrane successfully. Now we can see in our figure A that upon 3-day FEN treatment, there is more of the B band representing the core glycosylated protein, meaning there is more substrate for triple therapy to correct. While triple therapy treatment on its own for 1 day and 3-day show both bands, the greatest improvement can be seen in the 3-day FEN, 3-day triple group with zinc. Now one big question that remains is in patients, how effective is triple therapy or Trikafta treatment upon infection? So in these next few slides, I'll be giving just a little preview of some ongoing experiments that we are currently working on in the lab. To begin investigating this question, primary bronchial epithelial cells from a CF patient were used to look at electrophysiology of the cells with LPS challenge, which is a known virulence factor of Pseudomonas aeruginosa. Now, triple therapy effect is actually decreased by the LPS challenge, but in fact can be rescued by the addition of fenretinide. So given this improvement, we have designed a special tool that can monitor total CFTR protein and membrane CFTR protein levels using a high-bit tagged CFTR, which is in the 4th extracellular loop of the CFTR protein. An in-depth explanation on how this was done and how the tool works can be seen in poster number 668. So please take a look if that is any interest to you. In figures B and C, CFB41O negative overexpressing F508 del cells or epithelial cells with the introduction of the high-bit tag via CRISPR-Cas9 was used and measured using nanoglyluciferase lytic, which shows our total protein or total CFTR protein, and also extracellular, which shows our membrane protein systems. Now, in the presence of PA14 filtrate, triple therapy efficacy is actually lost. Treatment with fenretinide can prevent loss of responsiveness to the triple drug combination, and complete rescue of responsiveness is observed when adding LAU7B with triple therapy. So to put everything together, we'll look at some conclusions. So combination of triple therapy with LAU7B oral treatment significantly improved multiple markers of CF lung disease, including lung function and airway histopathology in the F508del TM1EUR mouse model. the combination therapy also improved several parameters at a cellular level, such as oxidative stress markers, fatty acids, phospholipids, and also protein levels. In primary epithelial cells, triple therapy effect is decreased by LPS challenge and can actually be rescued by the addition of phenretinide. And in CFB of 508del cells, the combination of triple therapy with phenretinide allows the rescue of triple therapy responsiveness, even in the presence of PA14 filtrate. Now, if we take all of these conclusions and put it together, the results in mice and in cells suggest a potential clinical benefit of the combinatory treatment of LAU7B and Trikafta in people with CF chronically infected with Pseudomonas and other lung pathogens. I would just like to thank everybody who's been involved in this project for all of their help with our collaborators at home and elsewhere, and I'd be happy to answer any questions. Thank you.
So we have one question from the audience. Can you comment on the reason for adding zinc in the Western blot data?
So zinc has been something that has been previously published by our lab in the past. So it shows that when we add it with phenretinide, it allows for more protein. Thank you.
What is your next step for these experiments? I know the mouse model is not always the ideal model for CF airway disease. Do you anticipate repeating these in any other models or just moving forward with the clinical trials that you described?
So our next steps would be to further complete the studies that I kind of gave a preview on, and to ensure that the data in vitro is solid so that we can move to our mouse model with various infections and to see how triple therapy works in both our in vitro and in vivo with various types of infections, and to take it from there and see how it goes. So. Yeah? Yeah, you can try. So that's a great question. It's definitely something that we are currently in the progress of trying to figure out. So far, we don't have a hypothesis, but with further experience, we're hoping we can figure that out and get back to you. Thank you.
Is there some thought that this could be also an add-on treatment for acute exacerbations, and is there any way for you to model that in the lab?
There are acute models. The model we work with, especially in vivo, is more chronic, but it's definitely something that we can look into in the future.
That would be really interesting because, we still have a good percentage of patients who don't recover lung function during their acute pulmonary exacerbations.
Thank you. Are we good? Thank you.
Thank you very much. And I think if there is no more comments, question, I think we will close this session. And I appreciate your attendance and activity. Thank you very much for coming.